Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Today, inshallah, we are going to revise the DNA problems. And to solve the DNA problems, you should remember some important points. The first type of problems is asking about the number of turns in the DNA double helix. And we have mentioned before, one turn equals or contains 10 pairs of nucleotides. So the number of turns equal the number of pairs of the nucleotides divided 10. When he mentions the number of nucleotides in single nucleotides, not in pairs, so we should divide over 2 to find the number in pairs, then divided over 10 or directly divided over 20 if you like. The second type is about the ratios of the nitrogenous bases. You know that adenine base pair with thymine, guanine base pairs with cytosine, so the number of adenine and thymine is equal, and the number of guanine and cytosine is equal, and the sum of them equal 100. So he may give us the ratio of one of the bases and ask about the ratio of the other one. When he asked about the nature of the sample, he mentions the ratio of 4 nitrogenous bases. Then he asks if this is a single-strand DNA, double-strand DNA, or RNA. Of course, if we find the adenine and thymine are equal, cytosine and guanine are equal, so it's a double-stranded DNA. If they are unequal, so it's a single-stranded DNA. If there is a uracil instead of thymine, so it's RNA. Now, the third type of questions or problems will be about the part of protein synthesis and RNA types. The number of codons of messenger RNA. You know that the codon is triplet. That means it contains 3 nucleotides. So the number of codons on messenger RNA equals the number of nucleotides divided 3. The number of amino acids equal the number of codons minus 1. This one codon is found at the terminal part of the messenger RNA, which represents the, of course, you know, stop codon doesn't code for any amino acid, so it is not counted. The number of amino acids equal the number of nucleotides divided 3, which represents the number of codons, minus 1 stop codon. Of course, you remember that the 3 stop codons are UAE, UAA, UAG, and UGA. Now, the number of amino acids equals the number of tRNA molecules. You know that the tRNA molecules, which they are the RNA type, which transfer the amino acids according to the codons found on the messenger RNA. Every amino acid is carried with a specific tRNA. So the number of amino acids equal the number of tRNA molecules. But if there is a repetition of codons, that means repetition of amino acids in the protein, so the number of tRNA molecules will be equal to the number of types of amino acids, not the number of amino acids. The number of amino acids, of course, they are equal to the number of anticodons. These are some numbers representing the diameter and the length, thickness of the DNA molecule. They may be mentioned in. Now, let's start answering the questions. We are going to start with the choose the right answer question. Question number 1. The maximum number of types of amino acids codes on the messenger RNA are, of course, you know that the total number of codes is 64, but remember that we have three stop codons, so the answer is yes, that's correct. 2. If there was a protein that composed of 300 amino acids, therefore the number of turns of DNA molecule that is transcribed to form this protein is how many turns? You know that 300 amino acids means that, yes, they are made of 300 codons, which are composed of 300 times 3, as the codon is triplet. So the answer is yes, that's correct. Number 3, if the number of nucleotides in one of the DNA molecules, not strands, molecules, was 270 nucleotides. Therefore, the number of formed amino acids are, of course, we have 270 um, nucleotides, so we are going to divide them over 2. Then we count, there will be 44 because we have one stop code. Question number 4. The least number of tRNA molecules needed to form a polypeptide chain that contains 50 amino acids of 15 types is, you know that we can count the number of molecules of tRNA according to the number of types, not the number of amino acids in case of repetition. So the answer is, of course, 15. To form a protein of 50 amino acids, the number of nucleotides that found on the messenger RNA must be at least. When he mentions the word at least, that means he is not counting the stop codon. Okay, pay attention. If he mentioned at least, so you will not count the stop codon. Okay, so there are 50 amino acids, that means 50 times 3. Okay, number 6, the least number of nucleotides in the messenger RNA molecule needed to form a polypeptide chain that is formed of 21 amino acids equal the least number of nucleotides in the messenger RNA, 63. Number 7, if the number of nucleotides found on messenger RNA is 45, the nucleotides, therefore the number of nucleotides in a portion or a piece of DNA molecule from which the messenger RNA is transcribed is, yes, 90. If the number of nucleotides in a piece of DNA is 300 nucleotides, therefore the number of amino acids that are produced after transcription of messenger RNA is 49. 300 divided 2 to count the number of nucleotides on one strand is 150. Divided 3 equals 50, but we have one stop codon. The codon which the release factor doesn't link with, of course, it's the AUG. UGA, UAG, and UAA are the stop codons to which the release factor links to end the translation process, as you remember. On measuring the percentage of nitrogenous bases of the nucleic acid was T equal 26%, A equal 20, G 23%, and 31% is the cytosine. So the nucleic acid is, as we have mentioned before, if they are equal, so it's a double-stranded. If they are unequal, so it's a single strand. And if there is uracil instead of thymine, so it's RNA generally. So the answer is a single-stranded DNA. Question number 2. If you have the sequence of nitrogenous bases in a piece of one strand of DNA molecule, runs as the following. Of course, we have to pay attention the single strand from which the messenger RNA is transcribed. It should start from 3' to 5'. So the question is, write the sequence of nitrogenous bases in the DNA that is complementary to the single strand mentioned previously. Okay, so from 5' to 3', and you know that guanine always placed there with cytosine and adenine with thymine. If there is a mutation, and what is the type of this mutation? Of course, the mutation occurs due to a change in one base, so it's a gene mutation. And of course, you know what is the effect of the gene mutation? It leads to formation of different protein, leading to different traits. If a sequence of nucleotides on DNA strand runs as the following. Number 1, write the sequence on messenger RNA. You should pay attention that it starts from 5', so we cannot transcribe messenger RNA from it. So we are going to write the complementary DNA strand, then the messenger RNA will be as the following. How many amino acids are formed and tRNA molecules that are responsible for the translation of the process? First question is the amino acids number. We are going to count every 3 nucleotides will be translated into one amino acid. It represents a codon for one amino acid. Of course, we are going to observe the last one, the last codon, the last 3 nucleotides. They are UGA, which is a stop codon, so we are not going to count them. So the number of amino acids equal 5. The number of tRNAs we have mentioned before. If there is a repetition, so the number of the tRNAs equal the number of types of amino acids, not the number of them. So we have a repeated codon here, AA. So the number of tRNA molecules is 4. If the sequence of nucleotides on DNA strand as the following, and the codons to of some amino acids are the following. Conclude the sequence of amino acids. Let's see. The first codon is GCT. No, this is not correct. We have mentioned first the complementary or the transcribed messenger RNA. Then we can find out CGA. CGA is the arginine. Then GCU is the alanine, and UGU is the codon of cysteine. Now, we have a messenger RNA molecule. What is the number of amino acids resulting from the translation of this messenger RNA? Let's count them. But you should observe the last one, UAG, so it's not counted. It is the stop codon, so the number is 4. How many types of amino acids? We have here a repetition. AUG is repeated twice, so the number is the number of types is 3. And what is the number of tRNA molecules? Of course, we have a repeated one, so it's not counted. So the number is counted according to the number of types, so it's 3 also. Number 7, if you have the sequence, as you see, write the complementary sequence. This is very easy, from 3' to 5'. Restriction endonuclease enzyme has a recognition site GAAT. You know that the recognition site is a sequence of nucleotides which the restriction endonuclease cut near or at it, leaving sticky ends. So, show by the arrows only the site of recognition on both strands. These are the two strands. Then, this is the recognition site, GAAT. So the restriction endonuclease will cut at or near it here. This is the first site. And of course, you remember that we read the recognition site from 5 to 3 on both strands. Number C, write the sequence of nucleotides in the resulted pieces after treated with the enzyme. Okay. A gene consists of 150 pairs of nucleotides. How many amino acids are used in formation of the resulted protein? 150 pairs of nucleotides, and you know that the gene is found on the two strands together. So one strand from 3' to 5', from which the messenger RNA is transcribed, contains 150 nucleotides. So the number of amino acids equals the number of codons minus 1. And you remember also the number of codons equal the number of nucleotides divided 3. So 150 divided 3 equals 50 minus 1 stop codon equals 49. Number 9. As you see, this is a single-stranded DNA from 3' to 5'. How many amino acids build the resulting protein? How many tRNAs are used in the translation process? Of course, first you have to transcribe the messenger RNA from this strand, and this is the result. Now, as you see, we have here UGA in the middle of the codons, and you know that UGA is the stop codon. So we are not going to count any of the codons come after the stop codon. So we have 1, 2, 3, 4, and 5. We have 5 amino acids. How many tRNA molecules? If there is a repetition, we are going to cancel 1. We are going to count them according to the number of types, so the number is 4. Assuming that the number of amino acids that form the different protein is 25, not 20, as you know, calculate the least number of nucleotides from which the genetic code can be formed. You know that genetic code is triplet. The code consists of 3 nucleotides to be 64, which is more than enough for 20. Let's see. I think the number is, yes, this is correct. The number is 3. So codon is triplet also if the number of amino acids is 25 instead of 20, except the region. Of course, when the codon is triplet, so they produce 4 power 3 equal 64 codons, which still more than enough for 25 amino. The last one, 16 types of amino acids participate in synthesis of a polypeptide that is formed of 250 amino acids. Mention number 1, the number of nitrogenous bases found on messenger RNA. Of course, the number is 753. as we have to multiply 250 times 3. It becomes, let's count it together. Okay, the number of codons is 251, and the least number of tRNA which is needed is 16, as we have 16 types of amino acids, and the last one is the type of bonds between the amino acids. Of course, it's a peptide bond. Peptide bonds attach or link between the amino acids together. Thank you very much, and till we meet again, inshallah, next time. Wassalamu alaikum wa rahmatullahi wa barakatuh.